Hi there, and welcome to A&P with Dr. O. In this video, we are going to walk through a sheep brain dissection. You'll complete a similar dissection in lab, and this will help us take a look at some of the different parts of the brain on an actual preserved specimen. The first thing that we're going to notice is that thick, tough outer covering of connective tissue that is going to be protecting the brain, and that is known as the dura mater. And we can see it here covering the cerebrum and then also covering the cerebellum here as well. I'm going to go ahead and turn the brain over so that the cerebellum and cerebrum are facing down. And then when I do that, and if I kind of move some of this excess tissue out of the way, I can find the pituitary gland. So right here, we can see the pituitary gland, and I'll go ahead and remove some of this excess tissue. That way we can see it a little bit better. As you can see, I've removed some excess tissue, including some of that dura mater. And again, we can see that pituitary gland a little bit better now. So that's going to be this rounded gland right here. And then we can also see some other structures, again, now that that dura mater has been removed. Once we find the pituitary gland, if we move upwards here, then we can see the optic nerves, optic chiasma, and optic tracts. And they make kind of an X. So if you see here this X-like shape that those structures make, we get the optic nerves are going to be on either side here as the top part of the X. And then the bottom parts of the X are the optic tracts. And where they meet in the middle of the X is the optic chiasma. So the optic nerves, this would connect out to the eye so that visual information could come in. And then the optic tracts allow that visual information to travel to the brain. Optic nerve, optic tract, and where they join in the middle of the X is the optic chiasma. We also can see the olfactory bulbs as well. And they are very prominent. They are much larger than the olfactory structures of humans. And that has to do with the differences in the range of sense of smell. So the sheep are going to have these larger olfactory bulbs, and that's going to allow for a greater sense of smell. But you can really see those, they're prominent, and you can see them on either side as we look at the inferior side of the brain. If we move away from the optic chiasma and the optic nerves and optic tracts, and away from the pituitary gland, then we can see some of the structures of the brainstem, and I'm just going to kind of push the pituitary gland off to the side so that we can see them a little bit easier. But what we have is we have the parts of the brainstem, and then we end with the spinal cord. So we would have the spinal cord, and then above the spinal cord is the medulla oblongata. And then we would have the pons, and the pons kind of makes this almost like belly. It kind of protrudes out like a belly here. And then above that is going to be the midbrain. So midbrain, pons, medulla oblongata, and then we are going to have the spinal cord. Now we're going to turn the brain back over and we're going to finish removing that dura mater. Now that we have all of the dura mater removed, we can identify the lobes of the brain. The names are going to match the bones of the skull that cover the lobes. So we're going to have the frontal lobes. Those are going to be covered by the frontal bone. We have the parietal lobes covered by the parietal bone. We have the occipital bone covering the occipital lobes. And then off on the side here, we would have the temporal lobe. We mentioned that this here was the cerebrum, and then this here is going to be the cerebellum. The cerebrum and cerebellum are separated by a fissure known as the transverse fissure. So I can put my pointer in here and we can see that transverse fissure. And then if I open up the brain, we can see some additional structures. So we have this structure right here that looks almost kind of like a little tongue-like structure. So that's going to be the pineal gland, which secretes melatonin. And then there are these structures here that kind of protrude out from the midbrain, and those are known as the corpora quadrigemina, and they process sight and sound reflexes. So we've got the corpora quadrigemina of the midbrain, and then just above that, again, this little structure that kind of looks like a little tongue there, that's going to be the pineal gland. Just a few more things to point out. When we are looking at the cerebrum, we have these raised areas that we can see that are known as gyri. And then we have these indentations, and those indentations are known as sulci. Again, the raised portion is the gyri, and the indentations are going to be the sulci. Also, on the cerebellum, if I flip the brain around, so we're focused now on the cerebellum, there is going to be a raised area in the middle here that separates the two cerebellar hemispheres, and that is known as the vermis. Next, we want to take a look at, inside the sheep brain. We want an internal view. So we're going to cut the specimen along this longitudinal fissure, which separates the cerebrum into hemispheres. So we'd have the right and left cerebral hemispheres separated by this longitudinal fissure. So we're going to go ahead and we are going to make a cut with a scalpel. Now we're looking at a sagittal cut of the brain, and we can see the corpus callosum right here, and the corpus callosum is what connects the two cerebral hemispheres. So now we can see that it kind of swooshes around here. And there's a space within the corpus callosum, and I can put my pointer into that space, and that's going to be the lateral ventricle. So that's going to hold cerebral spinal fluid. And then underneath, we have a space known as the third ventricle, which is associated with the thalamus here. So it's round and almost perfectly centered. And then just behind the thalamus is that pineal gland. So that little tongue-like gland that we saw earlier, now we can see it in sagittal section. Now below the thalamus is the hypothalamus. Hypo means below. So once you find the thalamus, then you know below the thalamus is going to be the hypothalamus. And then we can find the brain structures from this orientation as well. So we would have the midbrain, and then again, when the brain stem kind of makes a little bit of a belly, then we have the pons, and that's going to be followed by the medulla oblongata, and then that will eventually connect with the spinal cord. Now that the cerebellum has been cut in half and we have a sagittal section, we can see inside the white matter versus the gray matter. It looks like a tree, and it's called the arbor vitae, which means tree of life. So that's what we're looking at in this sagittal view here. So once we find the cerebellum and we find that space, there's the fourth ventricle, and we can trace our way back up to the cerebral aqueduct.